Good morning to all of you. I hope all of you are doing fine. So we'll be continuing from where we left yesterday. We were doing questions on uniform motion. First we'll finish one or two, three questions more, and then we will come to uniformly accelerated motion. This was the last question. I hope you all remember this was the last question. I told you the concept of average velocity, average speed and all, and I also told that a particular case when you see that the distances travelled in successive equal time intervals, if they give equal time intervals, when you break a journey into parts, then average velocity can be written as the average of individual velocities, means v1 plus v2 plus v3 plus v4 and so on divided by n, right? But if they don't mention equal time intervals, then you will go by the normal way. Total distance by total time gives you average speed, total displacement by total time gives you average velocity, right? I hope it is clear. Okay, so let's move to the next question. We can see in this particular question what they are mentioning is that point object traverses half the distance with velocity v0. Now we have a journey wherein, suppose we mark again this as AB. Now what they are saying is half the distance is covered with v0. So whenever you see half and all, means you can say that both parts are equal. Now if both parts are equal, so what I can do, I can take this as x, so this will also become x, both distances will become x. I hope it's clear. Now they are saying that first part you are traveling with a speed of v0, right, but the next half it is traveling with v1 for half time and v2 for rest half time. Now here they are using time, that means this journey again is split into two parts. So here you are traveling with speed v1 and here you are traveling with speed v2, and they are saying that both times are equal. So these are equal times. So suppose I take it as t, so this will be also t, both are equal. Now they are asking what is the average velocity for the whole journey. Clear? Average velocity. So what I can say is average velocity is total distance, which is 2x divided by total time. Now how will you solve this in a very simple way? How will you solve this in a very simple way? What we can do is, since this part, if you see from C to B, this part you can see the time intervals are equal. So I told you the method yesterday, if time intervals are equal, the average speed from C to B I can directly take as v1 plus v2 by 2 or not. These two are equal times. If these two are equal times, I can take the average as v1 plus v2 by 2. And what is the distance? X. Now let's write the average speed for whole journey. So when we go from A to B, the average speed for whole journey will be 2x divided by total time. Time from A to C and time from C to B. I can write time from A to C and time from C to B. So if I substitute 2x divided by, what is the time from A to C? Check in the diagram. X by v0, can I write it? And if I see the time from C to B, the distance is x and the speed from C to B is how much? v1 plus v2 by 2. So it will become v1 plus v2 by 2, 2 will go up. So now if I finally see x will cancel, take the LCM, v0 into v1 plus v2 upwards. So 2 is already there, v0 into v1 plus v2 will go up. Now what will be left below if you take LCM? If here already v0 is there, so we will get v1 plus v2, and if here we have v1 plus v2, we will multiply v0, so we will get 2v0. So simple way, the average velocity of the entire journey or average speed for entire journey, both will be same, because you are moving in the same direction without change in direction. So average velocity, average speed will be same. So here you can see that the final answer will be 2v0 into v1 plus v2 by v1 plus v2 plus 2v0. So yes, correct. Answer 3. Many of you are replying as answer 3, correct. Understood? So whenever you see that any particular part is split into equal time intervals, you can take the average speed of that part as the average of all the speeds. So V average from C to B is V1 plus V2 by 2, and for this part you already have V0. Now total journey average speed is total distance divided by total time. Please send your reply, yes, answer 3 is the correct one if you understood this. Okay, so that we can proceed to the next question. A boy goes from his home to school 5 kilometers away at 10 kilometers per hour. On reaching there and finding it closed, he instantly heads back, means he is not stopping, he is going from home to school, there he sees that it is closed, so instantly he turns back. And while turning back, he has also changed the speed. You can see 10 kilometers per hour, now he changed to 8 kilometers per hour. What is his average speed and average velocity magnitude in the first 45 minutes? They are not asking you, remember this thing, they are not asking you the total journey from home to school and school to home back. They are saying for the first 45 minutes. So we'll have to see how much distance did he actually move in these 45 minutes, right? So what we can do first is, if you again see, this is his home and this is his school, the distance is given 5 kilometers. Now when he is going, his speed is given as 10 kmph. So if it is given 10 kmph, so first of all, can we find the time, how much time he took from home to school? What will be the value? 5 kilometer divided by 10 kilometer per hour, means 5 by 10 is how many hours? 1 by 2 hours. 1 by 2 hours means how much? 30 minutes. Now, why am I taking this 30 minutes? The reason is, yes, correct, half hour, that is 30 minutes. Why I am taking so? The reason is that total time given is 45. So total time given is 45. So that means that I have to check how much time has already passed, so that I can see his further motion for rest of the 15 minutes. Home to school 30 minutes passed. Now how much time is remaining? Time remaining is 15 minutes. Remaining time is 15 minutes. Now convert it into hours. That is 50 by 60. How many hours? 1 by 4 hours. Is it so? So 1 by 4 hours is remaining. So in this, how much distance will he travel? So distance traveled further from school to another point. Let's he reaches a point P in 15 minutes. So what is his speed when he is coming? He is coming at 8 kilometers per hour. So when he comes at 8 kilometers per hour, so this distance will be 8 multiplied by how much time? 1 by 4. That is how much kilometers? 2 kilometers. Understood? Time is 1 by 4 hours and distance travel will be speed into time, that is 2 kilometers. Now, if you see the average speed is total distance by total time. Now what is the total distance? While going forward he travelled 5 kilometers and while coming back he travelled 2 kilometers, that is 7 kilometers. And what is the total time? If you see the total time was given as 45 minutes. What is 45 minutes equals to? 45 by 60, that means 3 by 4 hours. So if I write here 3 by 4 hours, so this will go up, 7 4s are 28 by 3, that is 3 9s are 27, 9.33 kilometer per hour. So average speed, if you check from here, this value is 9.33 kilometer per hour. Whereas if you see average velocity, we know it is displacement upon total time. Now yesterday I told you about displacement. Displacement, you are moving forward 5 kilometers and you are coming back 2 kilometers. So what is your displacement? if I take this direction, forward direction as positive, so 5 minus 2, whereas total time is still the same 3/4 hours, but displacement become 3. So when you write 3 by 3 cancels, 4 goes up, so average velocity becomes 4 kilometer per hour. So average velocity is 4 kilometer per hour. So which is now the correct option? If you see 9.33 and 4, that is option C. First we are moving forward, then backwards. Find the total time, total distance traveled, and if there is a change in direction, then we will say displacement will be forward distance minus backward distance. Is it clear to all of you? Let's move to the next one. Next question. These questions may be tricky, but remember, none of them are lengthy, none of them are very tough. You have to just think properly. That's what our purpose is. To understand the meaning of the question, try to draw diagrams. Everything will be clear and very simple. Next question is, a boy cycles from his village to the city and returns back in a total time of 2 hours. Now he is going from village to city. So suppose here it is his village and here we have city. But what they are saying is total time he takes is 2 hours. Now he maintains a uniform speed of 16 km per hour whenever the road is horizontal. Now we very well know, if you go on the roads while you are traveling in your town, in your village, wherever, you see that the roads are not always the same. Somewhere you will see the road will be horizontal, somewhere it will go uphill, and then somewhere it will go downhill, right? So we have a combination of different terrains, sometimes up, downhill, flat and all. Whenever the roads are flat, he can travel at 16 km per hour. Whenever the road becomes uphill, he can travel at 12 km per hour, and whenever 24 km per hour, whenever he is moving downhill, his speed will become 24 km per hour. So they are asking how far is the city from his village. So now what I can do is, village and city, the roads are a combination of uphill, downhill and horizontal, clear? So I can take all the horizontal portions, suppose this much. I don't know what is the length. Let's take this length as x kilometer. The horizontal portions in between village and city I am taking as x. Understood? Now all the uphill portions, these are the uphill portions, I am taking it as say y kilometer. I don't know its value. I am taking it as y. And all the downhill portions, I don't know. I am taking it as z. Okay. So x, y and z, these three distances, so they are asking the required distance between the village and the town or city is x plus y plus z kilometers. Understood? X plus y plus z kilometers. So now what happens when you have x, y and z? Now just see, whenever you are going, while going to the city, you are maintaining a speed of how much? They have given it is 16 kmph. When you are going uphill, this road is uphill, you are maintaining a speed of 12 km per hour. I am drawing the diagram. Diagram is only required. Rest everything you can very easily get. Let's see how. When you are coming downhill, your speed has become 24 km per hour. Please just reply fast. Did you understand this? The diagram I had drawn. I hope you all agree to this. Now this is the journey only from village to city. Okay. Now let's see the returning journey. Returning journey, can you say when you return the downhill journey, while returning what it will become? The journey z kilometer which was downhill while going from village to city. So if you go from city to village, the downhill will become what? Reply fast. Yes, uphill. And the uphill will become downhill. But the flat road will remain flat only. Yes. So see what happens. While returning, this becomes uphill. So now his speed will become 12. The uphill journey will now become downhill. So his speed will become 24 km per hour. And horizontal will remain horizontal only. This will be 16 only. So now if you see the total time he takes, what is the total time? Time is distance by speed. So while going x by 16, while going x by 16, while coming x by 16. Do you agree this? X by 16 going, x by 16 coming. Similarly, while going y distance your speed was 12, while coming back y your speed was 24. Similarly, while going your speed was 24 for z, and while coming back z distance your speed was 24. Please let me know this is clear or not. All the distance divided by speeds I have taken as time. Going, coming, going, coming. Okay. So once we write this, now see, if I take the LCM, what will I get? I will get x by 16, x by 16, x by 8, y by 12, y by 24. Take the LCM 24. This will also become y by 8. Z by 24 plus z by 12. Take the LCM, you get z by 8. Is it so? But what is the total time given in the question? The total time given in the question, you can see, is 2 hours. This time is given as 2 hours. So x plus y plus z divided by 8 is equals to 2 hours. So they are asking the total distance. So what will be this? It will be 8 into 2, that is 16 kilometers. So the distance between the city and the village will be 16 kilometers. That is option A will be correct. So the explanation, whatever I have explained, if you leave that, you just need to see that if you draw a diagram neatly, you quickly get, in one line you get the thing that total time, you write, and total time is given. So x plus y plus z, you get the value, that is the distance, 16 kilometer. I hope it is clear. Now one more question we will see on uniform speed, rest we will see then uniformly accelerated motion. Now in this particular question, Spider-Man, you can see he is at point A of a cubicle room. So cubicle room, at the point A he is sitting and he is deciding that he has to go from point A to point D. So from point A, which is the bottom corner, he has to go to the up corner, point D. Now they are saying that he should go there in the least possible time and he can maintain a constant speed v. So when speed is constant, now see, time we know is distance by speed. So if I want, now v is fixed, v is constant or fixed. So time will be minimum if distance is minimum. That is, he has to take the shortest path. He has to take the shortest path, then only he can reach the point D in minimum time. This is our only concept. But there are two ways. Number one, when he flies. Now when he flies by web shooting, he shoots his web and from A to D he has to reach. Can anyone say which path will he follow if he flies? What will be the shortest path? What will be the shortest path if he flies? Yes, it will be the body diagonal. If I see this point and this point, join them by the body diagonal, the straight body diagonal. So when he flies, shortest distance d minimum will be length of the direct body diagonal AD. Now you all know the formula, yes. Now this is given a cubicle room of length L. So body diagonal, if the lengths are L, the body diagonal will be under root of L square plus L square plus L square. How much it will be? Under root of L square plus L square plus L square, that is root 3L. So in this case, what is the minimum time? It will be root 3L divided by V. Very simple. Now comes the second case. Yes, first case is correct. Now comes the second case. Second case, when he crawls. Now if he is crawling, he can crawl on the floor also and he can crawl on the wall also. So his speed is constant. So can you tell over here the statement which I am telling is correct or not? Listen carefully. When he is crawling with the same speed on floor and wall, can I say that the floor and the wall are same for him? Can I say that the floor and wall are same for him? Krishna, it is wrong. AHD is wrong for second part. Listen carefully. So can I say that the floor and the wall will be same for him? Yes. So when he crawls, floor and wall same. Means either you draw the vertical or horizontal. So if floor and wall are same, instead of drawing vertical wall, I can make it horizontal also. So basically this floor and this wall, both are same for him. Both are same for him. So what I am doing, I am just drawing the shaded floor and wall. So what is the shaded floor and wall? You can see A, B, H, B, C. If you open this, so D and this point is G. Can you see? If I open this, then it will become this diagram. This is the diagram. I have made the vertical wall horizontal because it is same. Now, what is the shortest path from A to D? Can you see what is the shortest path from A to D? Now you can see this length L plus L, it becomes 2L. This length is L. So what is the shortest path? Direct diagonal is the shortest path. This is the shortest path from A to D. So if this is the shortest path, what is its length? Can I say in this case minimum distance will be this line AD, which is under root of L square plus 4L square, which is how much? Root 5L. It is root 5L. So if it is root 5L, what is the time taken in this case? Minimum time taken is root 5L divided by V. So second case answer will be root 5L by V. Yes. So when you are moving the same along wall and floor, you can take it as single. So it becomes a rectangle sort of HCD ABG. I have just made the wall horizontal. So now A to D, the shortest path will be this one. So if we select the shortest path, this is what we get as the answer, root 5L by V. I hope it is clear to all of you. So basically when you see it in the diagram itself, you will see that this is actually, we have split it into a rectangle. So actually he will start from A and on the floor he will be going up to somewhere here, and after this from here he will crawl up like this. Clear? So this red mark which I have drawn, this is the path which he will follow, and that is shown over here in this diagram also. Okay, fine. So now we are moving on to another concept which is we call as uniformly accelerated motion along a straight line. Uniformly accelerated motion along a straight line. Now till now we have been dealing with uniform motion where velocity was constant. Now we are talking about uniformly accelerated motion where what is constant? Acceleration is constant. Acceleration we know is a vector quantity, it is constant. Now I told you in last class, whenever something is constant, if any rate is constant, then its average value and instantaneous values both are same. Remember, again I am repeating, whenever we have any rate of change to be constant, then its average and instantaneous values both are same. So how we define instantaneous acceleration? Instantaneous, we have studied in calculus part, acceleration is rate of change of velocity, so dv by dt, this is called instantaneous, means at particular instant. And how we define average acceleration in an interval, you have studied this also. Average acceleration is called change in velocity by time interval. Change in velocity by time interval means v final minus v initial by time interval. These two definitions are given for instantaneous and average acceleration. Now if we use this concept over here, since acceleration is constant, so we can say average acceleration will be equal to instantaneous acceleration, and average acceleration is equals to instantaneous acceleration means delta v by delta t, both are same. So this value is constant. Now what does it tell me? If delta v by delta t is constant, means change in velocity will be directly proportional to time interval. Can I say? Change in velocity is directly proportional to time interval. Listen carefully. Change in velocity will be directly proportional to time interval if acceleration is constant. Means in equal time intervals, the velocity change will be equal. That is what we can say. In equal time intervals, velocity changes will be equal. So such type of motion, when acceleration is constant and the velocity change is directly proportional to time interval, we call such type of motion as uniformly accelerated motion. Now if we take initial velocity as u, final velocity as v, and the time interval delta t if I take as t. So suppose I go from point A to point B and we are moving with a constant acceleration from A to B, time interval is t. So what I just saw, acceleration and average acceleration both are same, so I can write from here acceleration will be change in velocity. What is the change in velocity you are seeing? It is v minus u. What is the time interval you are seeing? T. So from here we get a very important formula, v minus u is at, or we say v equals to u plus at. This all of you would be aware of. The expression that we are writing over here, v equals to u plus at. Yes, v equals to u plus at. This is the formula of finding velocity after a time interval t. Clear? Apart from this, if the distance AB we take as s, the distance travelled from point A to point B if we take as s, so you know the other important formula that comes by the method of integration is ut plus half at square. So the displacement or the distance both will be same in this case because you are not changing direction. So what is the displacement? Ut plus half at square. And when you make these two equations combined, so you get one more important equation which is v square equals to u square plus 2as. If you eliminate time from this equation, you substitute here, you get v square equals to u square plus 2as. Is it clear to all of you? These are the three important equations for uniformly accelerated motion. One is v equals to u plus at, one is s equals to ut plus half at square, and one is v squared is equals to u square plus 2as. I hope all of you agree to this. Now from this we get one more thing. Did I also tell many times what is the formula for average velocity? I am telling the trick. Let's see. Average velocity. What is the formula for average velocity? Tell fast. Which I told yesterday also. Average velocity is s by t or not? Average velocity is displacement by time interval. Yes. If average velocity is displacement by time interval, just divide s by t. If you divide s by t, see what you will get. You will get s by t, u plus at by 2, u plus at by 2. Now if you are getting u plus at by 2, take 2 as LCM. Take 2 as LCM. What are you going to get? 2u plus at. 2u plus at means u plus at. Listen again carefully. If I take the LCM, you get 2u plus at, and 2u can be written as u plus at. Now, what is the value of u plus at? Check from here. It is v. So in uniformly accelerated motion, very important and easy formula which you can use in questions. Average velocity, which is displacement by time. So displacement by time can also be written as u plus v by 2, means initial velocity plus final velocity by 2. So this formula can be useful in solving questions. We will see when s by t is u plus v by 2, it will give you answer in one line. This is a very important formula, and this result, change in velocity directly proportional to time interval. These are the two main results which act as shortcuts for different types of questions. Please note it, then you will see its application in a question, so that you will understand that yes, actually why it is easy. Okay, average velocity s by t is also equals to u plus v by 2 in case of uniformly accelerated motion. Okay, let's come to question. A particle accelerating uniformly along a straight line undergoes a velocity change from 3 to 13 in 4 seconds. Find its displacement during this time. Now, generally students will solve by which method? Let's see. Method 1. Whenever you solve questions on uniformly accelerated motion, the method that we generally use is, what you do is, you will take, suppose this point as A, this point as B. Initial velocity u is given as 3 meter per second. They have given the final velocity as 13 meter per second. They are asking this distance, and they have given the time. Time taken is 4 seconds. They have not given the acceleration. Now you will think, which is the formula which relates s, u, a and t? So you will be saying, okay, I will use s is equals to ut plus half at square. Now u is given as 3, time is given as 4, but acceleration is not known. So you will say 3 fours are 12 and plus 4 fours are 16 by 2, 8a. You will stop here. Now after that, what you will do? You have to find a. How to find a? We know acceleration can be written as v minus u by t, because v equals to u plus at. If acceleration is v minus u by t, v is 13, u is 3, t is 4. So 13 minus 3, 10 by 4, 10 by 4 means 5 by 2. So if I substitute in first equation, so s is 12 plus 8 into 5 by 2. 2 fours are 8, 20 plus 12, 32. So this is the main method which students use, and till class 10th they have been taught to do like this. But remember, if you see that acceleration is not mentioned, how shortcut you can apply if acceleration is not mentioned? Did I also say that average velocity is s by t is also u plus v by 2? Now, if you see, what is the time taken? 4 seconds. What is the initial speed? 3. What is the final speed? 13. So what is the value of s? 4 multiplied by 13, 3, 16 by 2. You get 32 meters in one line. In one line you are getting the answer. S is equals to 16 into 4 by 2, that is 32. Understood? So instead of writing two equations, just by a single equation you will get the answer. Is it simple or not? And simple and fast also, because when you will be doing objective questions in NEET, you won't be having so much time, so you can save a lot of time like this. Is it clear to all of you? Okay, now quickly one last question we will take, which again I will try to show you the tricks which I just told, so how we can do that question also. Okay, so I hope you have taken a screenshot of this, both the methods, right? One last question. A particle accelerating uniformly along a straight line undergoes a velocity change of 3 to 13. Again, same. You have a straight line. Your speed initially was 3 meter per second and it became 13 meter per second in 4 seconds. Acceleration is constant. Now they are saying find its velocity after next 12 seconds. So further you are taking 12 seconds, they are asking what is the velocity. Now again you have two methods. How can you solve it? Just see. First I can say from B to C can I write V equals to U plus AT? V is VC, U is from B to C initial speed is 13, acceleration is not known, time is 12. A is not known, again I will take A to B, again I will use V equals to U plus AT. This is the general method. Velocity is 13, U is 3, time is 4. So from here we get acceleration 5 by 2 meter per second square. Substitute here 13 plus 5 by 2 into 12, 2 6 are 12, 6 5s are 30 and 13. This is 43. Yes, answer D. But this is again a lengthy method. I don't like it. What is the simple method to do? If I go by the alternate method, did I say that change in velocity is directly proportional to time interval? Did I say this? Change in velocity is directly proportional to time interval. Okay. Now you see the time interval from B to C, is how much times the time interval from A to B? You can see 12 seconds is 3 times. Can you say? The time interval from B to C is 3 times the time interval from A to B. If it is so, then what I can say, that the change in velocity in BC is also equals to 3 times the change in velocity in AB. Now write the answer. If you know this trick, we can quickly say change in velocity in BC is VC minus 13. This is equals to 3 times. Change in velocity in AB is 13 minus 3. 13 minus 3 is 10. So you get VC is equals to 43 meter per second. So you can solve it orally if you just remember change in velocity is delta t proportional to delta t. So time interval becomes 3 times. So change in velocity will also become 3 times. If this is 10 meter per second change, so this change will be 30. If this change is 30, what was the speed here? 13. 13 plus 30 becomes how much? 43. So in any case, you are going to get 43 as the answer. I hope the points which I just mentioned are clear to all of you. So we'll be leaving the class now. Okay. So thanks to all of you for listening with patience. Okay. So we'll meet in the next class. Bye.
Yes, good morning everyone. Good morning. Is it audible and visible to you? Okay. So yesterday we started nomenclature, right? And we did some questions of alkane. And after alkane nomenclature, we started alkene and alkynes, right? Okay. So I told you 2 major rules for any nomenclature question. First is to select the parent chain. Okay. It is the most important rule. Select the parent chain. And second rule is from which end we should start numbering in this parent chain, right? So selection of parent chain and numbering of parent chain, right? This is the most important thing. Clear? So yesterday we did questions of alkene, right? So I'm giving you one or two question more, then we'll see some cycloalkane part also, okay? Look at this. Okay, try this. I'm giving question from, so carefully give the answer. Yesterday I ended the class at this point only. So please give answer very carefully. First of all, there is only one chain and we should start numbering from where? We should start numbering from which end? Left end or right end? I should start numbering from which end, beta? Please answer, left end or right end? Right, right. Very good. Why? Because if I start from here, I am getting 1, 2, 3, 4, 5, 6. 1, 6. From this end also 1, 6. But which one is more preferred out of double and triple? If both are giving us same numbering, then what is more preferred? Double or triple? Double. Double is more preferred. Yes. 1, 2, 3, 4, 5, 6, 7. Right? So name is what? Name is hept. Right? It's hept 1 in. What is coming, beta? 6 ein. Hept 1 in 6 ein. Very good. Very good. Okay. Next one I am asking. One more question. Just to clarify the yesterday concept. Now this time I will start numbering from where? Left end or right end? Please answer. From which end we will start numbering? Left end or right end? Right. Yes. This now I will not prefer double bone. Now so I will see location. From here I am getting first location. From here 1, 2. So I will start from here. Okay. Pi will be preferred over triple only when you are tying the rows. When the other rows are getting tied. Fine. Very good. So from here if I see bache, what is coming? Oct is what? Oct 6 in 1 ein, right? Oct 6 in 1 ein. And remember, if you see carefully, when I am writing in and ein together, I am not writing the e word of in. In case of in, I am writing en only. I am not writing e and e. When in ein is coming together, we are not writing the e and e. We are writing just en, right? Oct 6 in. E and E is not coming, just E and is coming, right? There are many rules you will study beta when you will deal in offline classes, okay? Okay, next one, your task is to find the main chain. Carefully give the answer. Your task is to find the main chain. Careful. Now this time you have to find the main chain. First of all, select the parent chain. Which chain I should take as my parent chain? Every chain is having pi bonds. If I take this one as my main chain, 2 pi bond. Here also I have 2 pi bond. Here also I have how many pi bond? 2 pi bond, right? So which chain I should take as my main chain, beta? See carefully. If I take this chain as my main chain, so this contains how many pi bond? 2 pi bond, or I will say 2 double bond, and this contains also 2 pi bond. This also have how many pi bond? 2 pi bond. But what is the benefit in this? Tell me why we are taking straight chain as our main chain. Tell me the reason. Because straight chain have 2 pi bond and longer length also. Now pi bond benefit though is present in all the chains, right? So I will take maximum pi bond and then I am going where? With the longer chain length. With longer chain length, right? So I can give the naming. Look at this. 1, 2, 3, 4, 5. 1, 2, 3, 4. Okay, so 1, 2, 3, 4, right? 5, 6, 7, 8. Done. And this time, this is our substituent. Carefully look at this. This is our substituent. This is a substituent, beta. Side chain, huh? This is a substituent. Right? Normally, a substituent with 2 carbon atoms, what we call ethyl, But this is not just ethyl, it is ethenyl because it have what? Pi bond here, right? So that's why the name of this substance is what? Ethenyl. Ethenyl. Yl is showing it is substrate. Yl word is showing it is what? Substrate, right? So what is the name for this compound if you write? Okay, the name will be, look at this beta, how many? Oct. First of all, write the substrate name, na? 4 ethenyl, substrate part. 4 ethenyl, the substrate beta. Next is what? The 2 pi bonds of octa. Octa 1 7 diene. Octa 1 7 diene. Most of you are doing very good. Fine. So remember here, this is our substituent. Assume by chance, you check bache, assume this is our main chain and this is our substituent. As I said, what is it? Ethenyl. As a substituent, what is it? Ethenyl. If I get this one, look at this. It is what? Propenyl. It is propenyl, right? Similarly, if I get this one, what is it? And one more thing you can mention here, it is better to give numbering to this also. I will say here, this is prop 1 enyl, right? What is it? It is, I can say, if this is my substrate, what is it? But 1 enyl. Why I am using yl word? Because it is now substituent. It is now substituent. Okay, bacha? Very good. Butynyl, propenyl, very good. Clear, bacha? Okay? So please give me answer to this question. Okay? Answer this. Carefully give the answer. This one, okay? And fine. Okay, beta. So please give me the name of this compound. Give me the name of this compound. First of all, tell me which one will be our main chain. Straight chain. Y state, beta. Y state, you can say maximum carbon. This also have same carbon. This also have what? Same carbon, right? Let me increase the carbon also here. Wait. Let me increase the carbon also. Let me make it little easy for you for now. Otherwise, many rules will come. Otherwise, many rules will enter into this question. Okay? This is my question. Otherwise, many other rules will enter in the question. So, please now look at this question carefully. Fine. Look, bacha, how many carbon here? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? And 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, right? Straight chain contains 2 pi bonds and is having what? Longest chain also. 2 benefits, right? So, can you give me numbering of this? From where I should go numbering? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Same thing. I'll start from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? So please tell me the numbering. Tell me the name of this compound. It is my what? Substituent. What do we call this? You can call just propenyl. Prop, or you can give the name propene 1I, like this one. Okay? It is for substituent. Where it is present, I will say on which carbon? 5 carbon. 5th carbon. What is present here? On 5th beta, what do we have? Prop 1 enyl. Right? Just to make sure it is 5 prop 1 enyl. Now which one? Next one? None. None. Nona beta. And now what is it? Next one? 1 8 diene. Fine. 1 8 diene. Is everyone? 5 prop enyl. 5 prop 1 enyl. Nona 1 8 diene. Why non? Why none? Because there are how many carbons? 9 carbon, because there are 9 carbon. Fine, bacha, right? Got the point? There are many rules which we have to learn step by step. Clear? If chain length becomes same, I added one more carbon in the chain, because if chain length becomes same, then numbering also matters. Okay? That's why I increase the chain length to make this as my main chain. Okay, bacha? Right. Done? So now you can see this is our name. Okay, bacha? So please, now we are doing some questions. Answer this. Okay, fine. First question is, select the structure with correct numbering. You have to select one with the correct numbering. Even you will see sometime when in a substituent we have many carbons, so we can give numbering there like 1 dash, 2 dash, 3 dash. Okay, that is also fine, but we will study all of these step by step, right? But tell me first of all, select the structure with the correct numbering. Is it right one? Is it right? Is it right? No, it is not right. Why? Because here triple bond, here which bond? Double. I should start from here now. 1, 2, 3, 4, 5. Because both are getting same location. Right? Very good. Is it right? No, I should start from here. 1, 2, 3, 4, 5, 6. Very good. Fourth. All of you are giving fourth answer, right? I will check beta. Why it is wrong? Why it is wrong, beta? Check carefully. It is 1, 2, 3, 4. And from here 1, 2, 3, 4. Here I am getting 1 to the pi bond and getting which one? 3. So 1 and 3 die in. In this case, what is coming? 1 and 4 die in. That's what is wrong, right? Now, next one is fourth one. Definitely it is right. Both are at same location, so definitely pi bond is more preferred, right? Fourth one. Very good. All of you are doing great. Giving right answer. Fourth one. Right, bacha? Okay, fine. Next one. Okay, write the name of this compound carefully. Give me the name of this compound carefully. Yes, bacha, if you write this carefully, what is it? C, double bond C here, this is C. This have 2 carbon upside, ethyl, here C. Now, what is my main chain? Our main chain can this one? Because it have more carbons. It have more carbon, right? What we will say now? What is it? 1, 2, 3, 4, 5, 6. So what is present here? At 4 we have methyl. 4 methyl. 4 methyl, hex, 1 ene. Okay, can you check what is coming? 4 methyl, hex, 1 ene. But third answer is right one. Third is right one. Some students are keeping one option. One is not right. Be careful. Be careful, because here you have more carbons in the chain. Here we have more carbon in the chain. Those students who are giving first answer, be careful. Don't make mistake. Here we have longer chain, na? 1, 2, 3, 4, 5, 6. Here how many? 1, 2, 3, 4, 5. It is my main chain. Okay? On this basis, I am giving one more question. Carefully give the answer. Because you did mistake. Okay? Look at this carefully. Tell me the name of this compound. They can write this way also, right? Assume here they are writing ethyl. C2H5, here also what? C2H5, and imagine here we have C3H8, or you can say 7, sorry. Carefully think about it. First of all, what is the meaning of C2H5? 2 carbons. C2H5, 2 carbons. C3H7 means how many carbons? 3 carbons. Carefully give the name of this compound. Don't forget, it is not 1 carbon, 3 carbons. Here, 2 carbons. Here, how many carbons? 2 carbons. Right. If I make structure for this compound, what is the real structure, beta? This is the real structure, na? C, double bond C here, C, and C. This upper side I have 2 carbons, in this side I have 2 carbons, and here I have 3 carbons, right? Clear? So now give me the answer. Choose the right one. Yes, bacha, choose the right one. Which is the main chain? What is the main chain? This is my main chain because it have more carbons with pi bond also including. Right? I'll go for this one. 1, 2, 3, 4, 5, 6, 7. At 4, what do we have? 4, 4 diethyl. 4, 4 diethyl. They are substitute, na, ethyl. And what is name? Hept1ene. 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 Yes, everyone? Right, very good. Be careful when they write C3H7 something, means there are 3 carbons. C2H5 means 2 carbons, here also 2 carbons. So don't make mistake here. I will take this as my main chain, okay? I will not take this as my main chain. Maybe they are writing C2H5, but how many carbons will come? C, okay? Very good. I hope you are clear now. Fine. Next one. Try this, beta. They are asking 2-ethyl, 3-methyl, hex 1,4-diene. 1,4-diene. Yes. Carefully use the answer. First of all, hexa. I am writing this one, huh? Hexa. 1, 2, 3, 4, 5, 6, right? Hexa 1,4-diene. Hexa beta 1, imagine this is 1, 2, 3, 4, 5, 6. 1 have ene and 4 also have what? Ene. Clear? Next one, what they are saying? 3-methyl, 2-ethyl, 2-ethyl, right? And 3-methyl. Is it the right structure? 2-ethyl, 3-methyl, hexa 1,4-diene. Which one is the right one? Tell me, beta. Very good. Out of the following, choose which one is right. Some are saying 3. Most of you are saying 3. We'll check. Very good. Fine. Okay, beta, let us check third. As you're saying, most one. It is the first carbon, right? First carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth. Very good. Right? Add 2, what do we have? Add 2, we have ethyl. Okay, add 2, beta, what do we have? Ethyl. And 3, what do we have? Methyl. Very good. Third answer is right. Ethyl means what? C2H5. Methyl means what? CH3. Ethyl and methyl. Very right. We will see next one. Okay, and we will start now cyclocompound because you know all these questions. You are giving right answer only. So we are starting the next point, nomenclature of cyclic compounds. Cyclocompound, right? Cyclo will use now. Okay, before that, one more you can give, right? Last question. Last question, beta. Find the answer for this question. IUPAC name they're asking for this one. So please make it fast. What is coming? What is my main chain? It is my main chain. Long chain including pi bond. 1, 2, 3, 4, 5. Add 2 we have what? Methyl. Add 3 we have what? Ethyl. Carefully give the answer. Most of you are saying 3. Okay, let us check third option. Let us check third option. 1, 2, 3. 3 ethyl. Yes, 2 methyl. And how many? 1, 2, 3, 4, 5. Yes, beta, pent1ene. They are writing 1 pentene or you can say pent1ene. Fine. Third is right answer. Very good. Very good. Why fourth is wrong? Why fourth is wrong? When there are 2 substituents, you will write substituents in their alphabetical order. You will write in alphabetical order. E will come first. In ethyl, E word will come first, then methyl M. Clear? Any my point? Here you are writing M before E. It is the wrong thing. And here to even numbering also wrong, na? Numbering also wrong given. And alphabetical order you have to follow. Ethyl E first, then which one? Methyl. Okay? Very good. Why 1 is wrong? Beta, 1 is wrong because I said na E should come first. E will come first and M will come later. In case of more than one substituent, you have to write the substituents in alphabetical order. Are we clear? I hope we are clear now. We have to follow alphabetical order. Fine, bacha? Okay. Now I am taking the next one, nomenclature of cyclic compounds. Okay, some question we will see. Cyclic compounds. Okay, bacha? So listen to me very carefully. We have done this part. I told you already. There is secondary prefix, primary prefix. There was word root, and it was you can say primary suffix and secondary suffix. Okay, and out of these, bache, which thing shows the cyclic word? This will decide your compound is cyclic or acyclic. For cyclic compounds, what we use, beta? We use cyclo word, cyclo. If your compound is cyclic compound, then you have to use cyclo word. That's the new thing only here. We will use cyclo word, right? Like this way I am giving example here. Imagine we have this thing. It is only one chain here, so I will say 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon, right? It's hexane, but not simple hexane. You say cyclohexane. You write cyclohexane, right? In cyclohexane, it is what? It is our primary prefix. Hex is what? Word root. And what is it? Ane is what? Your primary suffix. Primary suffix. Very good. Okay, bacha. Now please give me name of this compound. Give me name for this compound. Fast. If I am saying we are having this one, give me name for this one and name for this one also. Give me name for this compound. Fast. Count how many carbons are there and how many carbons here. Fast. Very good. Cyclopentane. Very right. This is cyclopentane. 5 carbon in the chain. I will use cyclo word to show it is cyclic compound and pentane. Cyclopentane. This one, beta, this one. Cycloheptane. Very good. It is cycloheptane. Very right. Clear? Now, next one is, look at this. If we have cyclic chain and we have it a side chain also, like side one, like look at this. If I have here one extra carbon, CH3. Now name this one. In cyclic chain I have 6 carbon, right? In side chain I have only 1 carbon. This is my main chain, parent chain. It is what? Substituent. It is my main chain, it is my substituent, right? Can you give me name of this compound? Substituent is methyl. Substituent is methyl. First of all, my main target is to find the main chain. First, our target is, okay, wait, don't message. Now, understand carefully. First of all, let's find the parent chain. Which one is parent chain? Side chain contains only 1 methyl, 1 carbon, and main chain, cyclic chain, have how many? 6 carbon. I will take 6 carbon chain as my main chain. I will take it as substituent. It is my substituent. Now, cyclic chain is my main chain. Cyclic chain is my main chain, and now I will start numbering. We know how to do numbering of parent chain. How? We have to give lowest locant. We have to give the lowest position to the locant. So I will start from here. Na? Look at this. 1, 2, 3, 4, 5, 6. First I am choosing my main chain as cyclic chain, and then I am giving numbering. So what is the name, beta? 1 methyl. It is 1 methyl. Then what will come? Cyclo. Then hexane. 1 methyl cyclohexane. I hope we are clear now. First select the main chain, then give numbering according to the rules we have done. there is no pi bond, there is no function group, so follow lowest locant rule. That's it. Okay, bacha? Very good. Now, give me name for this one. Careful. Give name. Okay, now tell me. Someone is asking, sir, why it can't be 2? Beta, when I can number from anywhere, na? In the cycle, I can start numbering from anywhere. So I will give methyl lowest position, means 1 position. Why I will give 2 position? When I can give first position, why I will give second position, right? Very good. Again, this is my main chain. Cyclic chain is bigger, na? Cyclic chain have 6 carbon. Side chain only have how many carbon? 2 carbon, right? Now, I will start from here. 1, 2, 3, 4, 5, 6. Okay? So 1 ethyl. Very good. 1 ethyl cyclohexane. 1 ethyl cyclohexane. Very good. I hope we are clear now. I hope we are clear now. Right? Very good. Now, next I'm taking. Okay, tell me this one. Okay, this one. Name this one. I should take which one as my main chain? I will take this as my main chain. 1, 2, 3, 4, 5, 6. Right? Clear, bacha? And I will start numbering from here. 1, 2, 3, 4, 5, 6. Clear, bacha? So what is the name for this one? It is 1 ethyl. 1 ethyl, 3 methyl cyclohexane. Cyclohexane. Fine, bacha? Okay, everyone. 1 ethyl, 3 methyl cyclohexane. Very good, very good. Very clear. Very clear, right? Okay, next one. Now, if you give me this right answer, then that means you know the nomenclature very clearly. Think carefully, give the answer, right? And don't make mistake. Try to be calm. Don't make hurry. Look carefully from where I should start numbering. Remember, lowest locant rule, then lowest sum. First of all, lowest locant rule, then what is important? Lowest sum of locant rule. Carefully, I will start from here. 1, 2, 3, 4, 5, 6. Because from here you see sum is coming less. Sum 1, 2, 4. So I will say 4 ethyl. The name is 4 ethyl. 1, 2 dimethyl. 4 ethyl. 1, 2 dimethyl cyclohexane, right? Cyclohexane, fine. Is everyone? 4 ethyl, 1, 2 dimethyl cyclohexane. Great, great, great. Okay, bacha, I am saying once again, if you choose this one, okay, some students maybe have doubt here. Look at this carefully. If you choose from here, beta, look carefully. Some students think, sir, why I can't start from here? If I start from here, 1, 2, 3, 4. What is coming locant position? 1 ethyl. What is coming? 3, 4. Location coming 1, 3, 4. Whereas in this case, what is location coming, beta? Here location coming 1, 2, 4. Which one is lowest sum, beta? Which one is lowest sum here? 1 plus 2 plus 4, how much? 7. 1 plus 3 plus 4, how much? 8. So we are following which rule? Lowest sum rule, right? If lowest locant rule tie, then we will see lowest sum rule. Lowest sum. Are you clear now? Are you clear this time? Why I am taking this one? Why I am not taking this one? Are we clear? Sure. Okay. Next one, if I say, now, if by chance side chain have more carbons, look at this. This is 4 carbon here. 1, 2, 3, 4, 5. If side chain have more carbon, then I will give side chain more priority. Side chain will be my main chain. Okay. So in such question, beta, look at this carefully. This is my first carbon, second carbon, third, 4, 5. This is our substituent. This time cyclic chain is our substituent and the side chain, outer chain is my main what? My main chain. So how I will name it? I will say 1 cyclobutyl. 1 cyclobutyl. It is our substituent, huh? It is my substituent. What is next part? Pentane. What is next beta? Pentane, right? Look at this carefully. This part is what is this? Secondary prefix. It is a word root. It is a primary suffix, right? Okay, everyone, clear? If side chain have more carbons, then take side chain as main chain and cyclic chain as substituent. Okay, are we clear? I hope we are clear. Okay, fine, bacha. Now look at this next one. Give me answer this question, bacha. If I have here 3 carbon cyclic chain and here I have 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. Tell me, what is the name of this compound? Write the name of this compound. Which one is our main chain? My main chain is my side chain, na? This one. 1, 2, 3, 4, 5, 6. It is our substituent. This is just our substituent. It's just our substituent, right? So please write the name of this compound. My substituent is what now? My substituent is 1 cyclopropyl. It is 1 cyclopropyl. It is my substituent. And what is main chain? Hexane, right? It is hexane. Clear, bacha? So you will see this is our substituent part. What do you call this substituent part? This is our this is our secondary prefix. This is a word root. This is secondary. This is primary suffix. Okay, one last question I am asking. So please answer it right. If we have this one, cyclic compound is having here pi bond and it is my methyl group. Last question. Carefully give the answer. The cyclic chain is our main chain. The main thing is from where I should start numbering. Be careful. As in numbering, my pi bond is more important. So I will start from where? Pi bond. I will say 1, 2, 3, 4, 5, 6. Okay, bacha. Next is 4-methyl. The name is 4-methyl cyclohex 1-ene. Cyclohex 1-ene. Very good. I hope we are clear about this. Remaining questions we will see next class. Okay. And I hope you are getting benefit from here. More rules, more detailed questions we will see in our offline classes. Fine. Okay. I hope these classes are giving you benefit. Remaining part we will see in next class. From tomorrow I think Jayansa will come. Okay. And continue the nomenclature part and remaining part like isomerism. Okay, bacha. Okay, bye then. I will see you then in campus only. Okay, see you in campus. Bye, bache. Okay, bacha, bye. Fine.
Hello, good morning, students. Good morning, all of you. Okay. Yesterday, you have learned some cell organelles, right? Which cell organelles you have learnt? Which cell organelles you have learnt yesterday? Okay, which cell organelles? Which cell organelles did you learn yesterday? Endomembrane system? Yeah, endoplasmic reticulum, very good. Yeah, butterfly lysosomes, very good. Golgi apparatus, yes. What about vacuoles? So now I'll ask you one question. Please answer to this question. Then we'll proceed to the next one. I have taught you yesterday endomembrane system. Endomembrane system. In the endomembrane system, how many cell organelles are present? How many? Number 1, 2, 3, 4. How many are present? Very good. 4, 4. Yes, yes. Very good. Yesterday we had discussed about 4 cell organelles in the endomembrane system. Yes. What is the first one in that? ER, Golgi complex and lysosomes, and then finally vacuoles. Vacuoles, right? We had discussed about ER, Golgi bodies, Golgi complex, lysosomes and vacuoles. Very good. Among these 4 cell organelles, among these 4, okay, which is concerned with protein synthesis? Among these 4, I gave you options already. ER, very good. Now, among these 4, which is concerned with packaging of the materials? Very good, Golgi. Very good, very good. Keep it up. Now, let us see among these 4, which are called suicide bags of the cell. Lysosomes. Very good. So, all of you know that. Correct, correct, correct. Very good. Good interaction. I should appreciate all of you. Can you tell me vacuoles are called as what? Storehouses of this cell or repository of this cell? Storehouses. Vacuoles are called. Tonoplast is the membrane of vacuole. Tonoplast. Last one question, then we'll proceed to the actual today's topic. Which vacuole is concerned with osmoregulation function? Is it a sap vacuole, gas vacuole, contractile vacuole, food vacuole? Contractile vacuole. Very good. So, contractile vacuoles are present in generally the protozoans like amoeba, euglena and paramecium. So, now I hope all of you have studied yesterday and you are answering to my questions. I hope you have done the reading of that particular things, cell organelles. Now, today let us continue with the cell organelles part 2. Cell organelles part 2. So, in this cell organelles part 2, you are going to learn about 3 cell organelles, particularly mitochondria, chloroplast and then ribosomes. So, first cell organelle is mitochondria. Let us go to that. So, all of you have seen that mitochondria in your cell topic already. Very good, powerhouses, correct. Let us see the structure first and then later we will proceed to the functions. Okay. All of you can see very clearly here, one particular shape is there for this. The shape will be sausage shape. Actually, different shapes will be there, but the shape, main shape is sausage shape or cylindrical shaped ones are present. So, the shape of mitochondria will be sausage shape or cylindrical shaped ones are there, and they are the cell organelles present in the animal cells as well as plant cells. They are present in both plant cells and animal cells. And this size of the mitochondria, generally it will be smaller than the chloroplast. This is smaller than the chloroplast. And this mitochondria is covered by 2 membranes. So, where outer one is called outer membrane and inner one is called inner membrane. So, there are 2 membranes around the mitochondria. The first one is, outer one is called outer membrane and inner one is called inner membrane. In between these 2, whatever the space that is present, it is called intermembrane space. So, you can think that this is the outer membrane and this is the inner membrane. And the space which is present between them is called intermembrane space. Intermembrane space. So, outer membrane is present and inner membrane is present and the space between them, the gap between them. You can see clearly in this we can see this gap. This is called as intermembrane space. So, can I say that mitochondria is having a double membranous envelope? It has two membranes around it. So mitochondria is having two membranes. So one is outer membrane, inner one is inner membrane, and in between those two membranes we have the intermembrane space. And if you see very clearly, the outer membrane is continuous. There are no interruptions for this outer membrane. Outer membrane is continuous. But whereas inner membrane, all of you can see here clearly, the inner membrane is showing the projections, finger-like foldings are there. The inner membrane. Inner membrane is not continuous because it shows some finger-like projections or infoldings. Okay. So this inner membrane is actually having it is not having continuity, but it will be having some finger-like infoldings. That infoldings are called as cristae. Infoldings of the inner membrane are called as cristae. Okay. So outer membrane is continuous, inner membrane is not continuous, and inner membrane shows finger-like projections or infoldings inside. That finger-like infoldings are called as cristae. And because of this inner membrane and outer membrane, okay, because of cristae, the mitochondria is divided into two compartments. This is called outer compartment, where I have written for intermembrane space, that is only called as outer compartment, and this inner one is called inner compartment. Inner compartment. Okay, so two compartments are there because of the outer and inner membrane. So the inner membrane inside, whatever the compartment is there, okay, that is filled with different materials and it is called as matrix. Inside this blue color one, whatever this blue color one is there, inside the inner membrane, it is called as matrix. It is filled with all the respiratory enzymes. Different kinds of respiratory enzymes will be there in this matrix. Not only respiratory enzymes, it also has ribosomes, particularly 70S type, and then some DNA will be there, some granules also will be there, some circular DNA will be there, and RNA also is present. DNA, RNA also is present in this. So inner compartment is filled with matrix. It is made up of different things. Matrix contains mainly respiratory enzymes, okay, which are participating in the aerobic respiration. Then it also contains one double-stranded DNA. It is circular. It will be looking like this and it will be having two strands and it is circular in nature. One single circular double-stranded DNA is present. Then few RNA molecules. They are actually single-stranded ones. Like this they will be there. DNA will be looking like this. This is the double-stranded DNA which is present, circular DNA present in the mitochondria. And like this, RNA molecules are present, which are generally single-stranded RNA molecules. They are present inside. Along with that, we have ribosomes. The ribosomes will be looking like this structure, and the type of ribosomes are 70S type of ribosomes. These three, along with the respiratory enzymes, are present in the matrix. Respiratory enzymes. Okay. What and all are present in the matrix? First, respiratory enzymes, then a double-stranded DNA which is circular in nature, then few single-stranded RNA molecules, and then few 70S ribosomes. These are all present in the matrix which is actually filling up the inner compartment. So in the inner compartment, matrix is present with all these. Okay. Outer compartment is not filled with anything. No liquid will be there generally, and it is not having anything, but it will be having some enzymes which are associated with that. Okay, inner membrane will have its own enzymes, outer membrane will have its own enzymes. Outer membrane is permeable, inner membrane is selectively permeable. So it will allow everything, but inner membrane is less permeable. So that permeability also differs. Each membrane will have different respiratory enzymes. But in the space, generally protons will accumulate. Only protons can be there in this. Ions, H plus ions only can be there in this. This is about the mitochondria, and you can see in this structure you have learnt about all the parts except ATP synthase particles. ATP synthase particles. You can see here ATP synthase. So, ATP synthesis occurs on the inner mitochondrial membrane with the help of that ATP synthase enzyme. So, ATPs are synthesized in the matrix, particularly on the inner mitochondrial membrane. So, these ATPs are synthesized here and they are also stored here. As the mitochondria is involved in the synthesis and storage of ATP, these mitochondria are called as powerhouses of the cell. What is their nickname? Powerhouses of the cell. Okay? They are called as powerhouses of the cell because they are synthesizing ATP. Energy currency of this cell will be synthesized by this, and they are storing that ATP for the future use also. So, as they are synthesizing the power, they are called as powerhouses of the cell. Okay. Their shapes are sausage shape, cylindrical shape, anything can be there. Then they have two membranes, outer membrane, inner membrane, and the space present between them is called intermembrane space. Okay. And it divides, these two membranes will divide the mitochondria into two compartments, outer compartment this one and inner compartment this one. Inner compartment is filled with different things, homogeneous matrix. It contains respiratory enzymes, one double stranded DNA, few RNA molecules and few 70S ribosomes. And that inner membrane infoldings will be there, they are called as cristae. On that ATP synthase particles will be there, and that ATP synthase particles will be synthesizing the ATP. As it is synthesizing the ATP, adenosine triphosphate, these are called as powerhouses of this cell. I hope all of you understood. Let me continue with the next cell organelle. Before going to that, I would like to ask you one question. In mitochondria, how many membranes are there? In mitochondria, how many membranes are there? Two membranes. Very good. What is present between outer membrane and inner membrane? What is it called as? Between outer membrane and inner membrane? Intermembrane space, right? In mitochondria, how many compartments are there? Two compartments, outer compartment, inner compartment. Very good. Finger-like infoldings are there in the inner membrane. They are called as, very good, cristae, cristae, very good, very good. What lies in the inner compartment? What is present? What is present in the inner compartment? Matrix, very good, matrix, correct. In which area you can see the ribosomes? Matrix, intermembrane space, inner membrane, outer membrane. Matrix, correct. So let me proceed to the next cell organelle, that is chloroplast. Chloroplast. Okay, very good, very good. So mitochondria has two membranes, double membranous bound cell organelle. In the same way, chloroplast also has the double membranous envelope. You know, their outer membrane and inner membrane is there. Even here also outer membrane and inner membrane is there. But here the main difference is between the membrane space is also present. It is called intermembrane space, you can call, or periplastidial space. The only difference is both the membranes are continuous here, whereas in mitochondria only outer membrane is continuous, inner membrane shows infoldings. Here both are continuous. You cannot see any kind of infoldings here. You can't see. So outer membrane and inner membrane both are continuous. So you can see here internal membrane, okay, external membrane, nothing but outer membrane, inner membrane. The space present between them is called intermembrane space. Already you have learned that same thing. But here the point is both the membranes are continuous. Outer membrane is permeable, inner membrane is selectively permeable, little bit less permeable. Outer membrane is almost more permeable than the inner membrane. That point is applied for both the mitochondria and chloroplasts. So inside this inner membrane, what is present is this total thing. Okay, the empty area, all of you can see this empty white color area, it is called stroma. So in the inner compartment, we can say that outer compartment is not having anything. In the inner compartment, but you are not supposed to use that inner compartment. We can say inner membrane is limiting. Okay, inner membrane is limiting. It is a boundary for stroma. So what lies inside the chloroplast, in the inner membrane, inside the inner membrane is stroma. Okay, what is present on the inside of the inner membrane? Stroma. In the stroma, you can see this white one is a stroma, and in that small green color structures are present. These green color structures. These green color structures are called thylakoids. Okay, each one is called as thylakoid and they contain only one membrane. Each thylakoid will be having one membrane. So in the stroma, these green color structures are there. That green color single membrane bound sacs, they are called as single membrane bound sacs are there. They will be like this. They are called as thylakoids. And this empty cavity in them is called lumen. And this thylakoid, if they are stocked one above the other, like pile of coins, it will form one granum. Grana plural. Okay. So in the stroma, we have single membrane bound sacs which are called as thylakoids. And thylakoid inside, they have the cavity. That cavity is called as lumen. And the thylakoids are arranged one above the other like a pile of coins. That pile of coins will form the granum or grana. And on this thylakoid membrane, we have pigments. You know, chlorophyll pigments are there. That pigments are present here, chlorophyll, which are concerned with trapping of sunlight energy for the photosynthesis. So on the thylakoid membranes, we have the pigments like chlorophyll and they'll trap the sunlight energy, and with that energy, these molecules will be involved in the photosynthesis process. You know, photosynthesis means it will be synthesizing the food materials like glucose. So the thylakoids will form the grana. They are actually present in the stroma. But now in the stroma, all of you can see in the stroma, chloroplast DNA is there. Chloroplast DNA. This purple color one is there, that is a chloroplast DNA. Like the mitochondria, this also has a double-stranded circular DNA. It is circular because the ends are connected. You can't see the circular nature very clearly here, but you should remember this is circular double-stranded DNA. And apart from that, you have this few RNA molecules will be there here, like the mitochondria, and we have even ribosomes here, 70S ribosomes. Plastoglobule is nothing but one kind of storage material. Okay, here we have even the starch grains also will be there. Proteins also will be present. Certain kind of proteins also will be present here. Okay. Now, if you can see here, grana or connected by, grana or interconnected by, you can see that this green one is there. They are all connecting the grana here. These are called stroma lamellae. They are called as stroma lamellae. Okay. So, this stroma lamellae will be connecting the two grana together and they will be in contact with the stroma. And inside this stroma only, we have all enzymes which are required for photosynthesis. So, stroma, like matrix, it contains photosynthetic enzymes. Because it is involved in photosynthesis, only photosynthetic enzymes are present in the stroma. And one double-stranded DNA will be there, chloroplast DNA. Circular DNA will be there, that is called chloroplast DNA. And then few RNA molecules are present like mitochondria. 70S ribosomes are present like mitochondria. Here they contain the photosynthetic enzymes. In mitochondria, they contain the respiratory enzymes. That is the main difference. Okay. And in the stroma, we have different enzymes like rubisco. One major enzyme which is present in the stroma is rubisco. This is concerned with carbon dioxide conversion to glucose. Like that, so many enzymes are present. Okay. So, what is a major function that is performed by the chloroplast? You can see here, the basic structure will be looking like this. Isn't it looking like our lens, eye? So the shapes are different shapes are there, like lens shaped, oval shape, disc shape, okay, ribbon shaped. This many shapes are there for the chloroplasts. And even number of chloroplasts also will be different from one type of cell to another type of cell. You know, this is the outer membrane, this is the inner membrane, and this is intermembrane space. Already I have given you the names there, so I am using the shortcuts. Outer membrane, inner membrane, intermembrane space. And this entire area inside the inner membrane limits the stroma. Inside that stroma, we have a double-stranded chloroplast DNA. Chloroplast, circular double-stranded DNA. And inside we have few RNA molecules. And we have 70S ribosomes. 70S ribosomes. And different enzymes, starch granules like that, they will be there inside this. But again in this, we have thylakoids. Each one is a thylakoid. Okay, and these thylakoids are connected by, okay, stroma lamellae. These are called stroma lamellae. These are called as grana, granum, and this is thylakoid. One only I have shown you there, so thylakoid. Okay, so these are larger than mitochondria. Definitely these are larger than mitochondria. These are present only in the plant cells, and some algal cells these are present, which are involved in photosynthesis. In that cells only these are present. So mainly when you take plant and animal cells, in plant cells these are present, okay, not in animal cells. So which cell organelles are absent in animal cells among mitochondria and chloroplasts? Chloroplasts are absent. These are particularly unique features of plant cells. So this is the actual structure of the chloroplast, which are having different shapes like lens shape, oval, disc, ribbon shaped, etc. But mitochondria shapes are sausage shape or cylindrical shape. What is the function of this chloroplast? Chloroplasts are concerned with photosynthesis. So these are called photosynthetic cell organelles. These are called as photosynthetic cell organelles or kitchen of this cell. Kitchen of this cell, because they are concerned with production of glucose. Glucose is nothing but food. Okay. So this is involved in photosynthesis. So it is called as photosynthetic cell organelle. That is the main function of the chloroplast. Okay. Mitochondria are involved in respiration. So they are called as respiratory cell organelles. They contain respiratory enzymes. So they are called as respiratory cell organelles. Whereas chloroplasts, they contain photosynthetic enzymes. So this is called as photosynthetic cell organelle. Because it is participating in photosynthesis, this is called as photosynthetic cell organelle. And as it is preparing the glucose, which is a food for all the organisms on the earth, it is called as, this cell organelle is also called as kitchen of the cell. Both the membranes are continuous. I hope all of you understood chloroplast. Yes, very good, very good. Any doubts in the chloroplast? Yeah, both are same, but granum is singular. Singular, one. Okay, one which is looking like this. If you write so many grana, that means 2 grana if you write. This is now called as grana, more than one. Pile of coins. How is it involved in metabolic synthesis? Not metabolic synthesis, it is involved in photosynthesis, food material. Did you learn the equation of 6CO2 plus 12H2O gives rise to light energy chloroplast, glucose and oxygen? Glucose and oxygen, photosynthesis reaction. The photosynthesis reaction will be like this. 6CO2 plus 12H2O gives rise to light energy, chlorophyll pigments which are present on the thylakoid membranes. They'll form the glucose, C6H12O6. This is glucose and it yields 6 oxygen molecules and 6 water. This reaction is called photosynthesis reaction. So chloroplasts are involved in this. Chloroplasts are involved in this. Okay. I hope all of you understood now. Let us go to the next cell organelle, that is ribosomes. The next cell organelles are ribosomes. These are made up of particularly RNA and proteins. Which type of RNA will be there? rRNA is present. RNA means ribonucleic acid. Okay, Swati, do hear ma? RNA is nothing but the ribonucleic acid. So, rRNA is present along with proteins to form the ribosomes. How many membranes are there in the mitochondria and chloroplast? 2 membranes. Do you remember? So, now ribosomes are made up of only 2 things. One is rRNA, ribosomal RNA, ribonucleic acid, and proteins. And here, can you see any membranes? No membranes. So, very, very important point here. This is amembranous cell organelle. Amembranous means no membrane. Non-membranous also you can call it. Non-membranous or amembranous cell organelle. This is present in both prokaryotes and eukaryotes. In the first class, I have shown you the only one cell organelles which are present in prokaryotes are ribosomes, right? So, these are present in both prokaryotes and eukaryotes because they don't have any membrane around them. There is no membrane around this. So, any of the ribosome, it will be looking somewhat like this with a larger subunit and a smaller subunit. Okay, so ribosomes are made up of basically rRNA and proteins and they contain 2 subunits, 1 and 2. This looks small, no? So, it is called smaller subunit. This is larger, so it is called larger subunit. So, both larger subunit and smaller subunit, they will join here exactly in the middle and it will form a complete ribosome structure. And these ribosomes, that subunits I have written smaller subunit and larger subunit. In prokaryotic cells, the 70S ribosomes are present. In prokaryotic cell, which type of ribosomes are present? 70S type of ribosomes are present. Whereas in eukaryotic cell, 80S type of ribosomes are present. You can call this 80S type of ribosomes as cytoplasmic ribosomes. Whereas 70S ribosomes are called as organelle ribosomes or prokaryotic ribosomes. Because these are present in the 70S ribosomes are present in the mitochondria and chloroplast. So, they are present in the mitochondria and chloroplast, so they are called as organelle ribosomes. And they are the only one type of cell organelles present in the prokaryotes, so they are called as prokaryotic ribosomes. So, prokaryotic or organelle ribosomes are 70S, whereas eukaryotic and cytoplasmic ribosomes are 80S. So, in the eukaryotes, particularly in the cytoplasm, 80S ribosomes are present, and in the prokaryotes, mitochondria and chloroplast, 70S ribosomes are present. Here, the subunits you can see, the smaller subunit in 70S is 30S, larger subunit is 50S. Here the smaller subunit in 80S is 40S and larger subunit is 60S. When you add these two, you will be getting 80s particularly, but there is an overlapping zone. They will be overlapped here. So due to overlapping, it will form only 70s. If you add this one, you will get 100, but you will be getting only 80s because of the overlapping between the smaller subunit and the larger subunit. Now let us come to the S. Riyan, concentrate. S. What is S? I have been using that S. What do you mean by S? S stands for, S means Swedberg unit. It is a unit. Okay. Whatever the 70s or 80s we are writing, S is nothing but Swedberg unit. It represents, it will give us the sedimentation coefficient. Sedimentation coefficient. Okay, at how much rate the ribosomes are sedimented. When you keep the cells, cell organelles in a test tube, and if you subject them to centrifugation, then they will be sedimented at a particular rate. You will get to know after coming here what is this. So S means Swedberg unit and it represents the sedimentation coefficient by which we can measure the density and size of the ribosomes indirectly. It gives indirectly a measure of density and size of the ribosomes. So small ribosomes are 70s ribosomes, larger ribosomes are 80s ribosomes. 70s are present in the prokaryotes, mitochondria and chloroplasts. 80s are present in the cytoplasm of eukaryotic cells. So organelle ribosomes and cytoplasmic ribosomes. I hope all of you understood S, Swedberg unit. It represents the sedimentation coefficient which actually gives us indirect measure of density and size of the ribosomes. The last one, the function of ribosomes. They are involved in protein synthesis. So ribosomes are called as protein factories of this cell. Protein factories of this cell. Okay. So what is the ribosome function? Protein synthesis. As they are involved in protein synthesis, they are called as protein factories of this cell. Okay. If you have any doubts, you can ask here. What is the purpose of the DNA and RNA? Yes, the DNA and RNA, they will synthesize the chloroplast and mitochondria proteins. The proteins which are required for the chloroplast and mitochondria, they will be synthesizing that partially. And these two, they will divide by fission without depending on the nucleus for their division. So mitochondria and chloroplast, they have their own DNA. No need for them to depend on the nucleus. So chloroplast and mitochondria are called as semi-autonomous cell organelles. Vacuole is absent in animal cell. No, vacuole is present. Small vacuoles are present. Larger vacuoles are present in plant cells. What is meant by prokaryote? Which is having the primitive nucleus. There is no nucleus particularly. It is not surrounded by any nuclear membrane. It is not having nucleolus. A primitive nucleus will be there with just one chromosome. No membrane around it. That is called prokaryote. Okay. I hope all of you understood. Yes. Okay. All of you prepare these two cell organelles, particularly mitochondria and chloroplast, and ribosomes anyhow it is smaller one. So tomorrow we'll continue with the next cell organelles. Thank you girls, guys. Thank you. Bye. Bye ma.
Hello everyone, good morning all of you. How are you all? Start today's class without any delay. So yesterday I have told you about ecology, isn't it? I have told you about ecology. And in the ecology I have told you now we have to study only 3 chapters. Yes or no? We have to study only 3 chapters, beta. And the very first chapter is organisms and population. Organisms and populations. All right. So I have told you yesterday few things. Yesterday you all have understood what is the meaning of an organism, What is the meaning of a population? What is the meaning of a community? What is the meaning of ecosystem, right? What is the meaning of ecosystem? What is the meaning of biome? And what are the 4 levels of biological organization? Everyone. So the 4 levels of biological organization were organism, population, community and biome. Yes or no, everyone? So tell me, have you understood this much? Yesterday only I have given you the glimpse of the ecology, the brief introduction about the ecology, what you have to study, what you don't have to study. I have shown you the comparisons between the old and the new NCERT, and I have told you all these terms, beta. Yes or no? Yes, everyone have understood very well. All right. Today, I am going to introduce you another topic that is also the part of this chapter only, beta. This is the chapter Organisms and Population. In this chapter only we have a very, very interesting topic, and that topic is you must have come across, right? You must have come across this topic. You must be knowing this topic already. So what is it? What we are going to discuss now in this chapter? Yes, everyone. Population interactions. Yes, very good. Population interactions. So as you know what is a population, now I will be telling you population interactions. Okay. So when I am interacting with you, you are interacting with me. Is this a population interaction? No, beta. Because we belong to the same population. When different populations are interacting, then you can say that this is a population interaction. Okay, so here in these population interactions, we are studying interspecific interactions. We are studying, beta, inter means between, species means different species. So interspecific interactions. We are going to study interspecific interactions, beta. Okay, interspecific interaction. So let us suppose I have a dog. You know that I don't have dogs and I don't love dogs. But still let us suppose that I have a dog. Now I am giving him food. Yes or no? I am giving food to the dog. Dog is a different species. I am a different species. So we are interacting. I am giving food to the dog and dog is giving me protection. It is taking care of the house. Yes or no? If any unknown person will come, we will be aware already. Yes or no? So we both are getting benefit from each other. This is a population interaction, beta. This is a population interaction. So you must have seen many population interactions in your nature, around yourself. If you will observe the surroundings, na beta, if we interact with dogs, then it is population interaction. Yes, beta, yes. If we interact with dog, this is a population interaction. If we interact with cat, this is a population interaction. If cat and rat are interacting with each other, if cat is eating the rat, you know, this is population interaction. Okay, everyone. Yes, everyone. Now tell me, if you are eating chicken, yes, it is also a population interaction. If you are eating chicken, I don't eat chicken, but I know that most of you people are eating chicken, yes? It is your favorite, butter chicken, butter naan. I know that. Okay. So if you are eating chicken, this is also population interaction. Chicken is a different species. You are a different species. Okay. So let us suppose if I am not eating chicken, but I am eating a different vegetables. I am eating salads. I am eating like different grains, rice, chapati. This is also population interaction. Do you know that? So population interaction is between the different species, Alright, humans and plants. Yes, everything is interaction, beta. Between humans and plants also interaction is there. Plants are getting carbon dioxide and we are getting oxygen from the plants. This is also an interaction. So wherever interaction will not always be beneficial. In the interaction there can be 3 kind of things, okay? So in case of population interaction, the one species can be beneficial. or beneficial. It is getting the benefit. It can be harmful also. Harmful also. Okay, harmful, or you can say detrimental. Detrimental. Okay, let us suppose a cat is eating the rat. So the rat is being eaten in this interaction. Rat is being harmed. Cat is getting the food, but rat is being harmed. So one is getting benefit, other is getting harm. Okay. So sometimes both the species will get the benefit. Sometimes both the species will get the harm. Sometimes one species will get the benefit, other species will get the harm. And sometimes it is neutral, beta. Neutral, neutral, neutral. Okay. It is neutral. That means it is not even getting benefit and it is not even getting the harm. It is neutral. So let me show you 6 types of population interactions. Yes or no, everyone? Can I move ahead? Have you understood the meaning of population interaction? Population interaction is the interaction between different species. If lion is eating deer, that is an interaction in which one is getting the benefit, one is getting the harm. I will tell you all these types of interactions with examples. I told you na, today you are going to see beautiful pictures and you are going to enjoy a lot in my class today. Okay. So let us start. Let us go to the next page. Here I have written 6 types of interactions, beta. What is the first type of interaction written here? Mutualism, mutualism, mutualism. Mutual. Mutual means both are getting benefit, beta. Plus means benefit. Beta, plus means benefit. Plus means benefit. Okay. Plus means benefit. Plus means benefit. Minus. Minus means what? Minus means, beta, minus means it is harmful. It is getting harm. Okay. And 0 means what? 0 means what? Whenever there is 0, 0 means it is neutral. It is not benefited. It is not harmed. So now look here, everyone. First interaction is mutualism. You must have heard of mutualism. Yes or no? You must have heard of mutualism in your surrounding. Pollination. Yes or no? Do you know pollination? Pollination is a mutualism. The pollinators are getting the nectar. है ना? The pollinators are getting the food from the flower and the flower is getting pollinated. Yes or no? So do you know what is pollination? Everyone in the botany you must have studied pollination. I know you have studied pollination. So both are getting benefit. Just like that, I will tell you more examples of mutualism. Okay. Competition. What is among yourselves? You all are competing with each other. Okay. You all are in competition with each other. Competition is that kind of interaction in which both will be harmed. Let us suppose 2 kids are fighting. Obviously, this is not a population interaction, but I am just giving you an analogy. I am just giving you the example. Okay. 2 kids are fighting. So both of their mothers will scold them, na? Why you are fighting with him? Why you are fighting with him? Both will get the scolding. Both will be harmed. So competition, remember, competition is the interaction in which both will be harmed. Both will be harmed. Okay. Predation. Predation. What we are doing? Predation. When we are eating chicken, predation. When lion is eating deer, predation. When lion is eating any other animal, predation. When I am eating paneer, predation. When sparrow is eating seeds, predation. So predation is that kind of interaction in which one will get the benefit. Who will get the benefit? Beta, predator will get the benefit. Predator. If we are eating chicken, we are predator. We are predator. Okay. And the one who is getting the harm is beta prey. 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 So, if I am eating chicken, let us suppose my prey will be chicken and I will be the predator. If lion is eating deer, lion will be the predator and deer will be the prey. Alright, so have you understood what is the meaning of predator and prey? Predator is getting the benefit and prey is getting the harm. Prey is being eaten by the predator. Prey is being eaten by the predator. Yes, everyone, tell me quickly, tell me, have you understood prey and predator? Have you understood prey and predator? Now, next is parasitism. Parasitism. These both are same, beta. They are same. They are same. One is getting benefit, other is getting harm. So parasitism also, one is getting benefit. Who is getting benefit? Parasite. Parasite is getting the benefit, beta, and host. Host is getting the harm. Host is getting the harm. Do you know what are parasites? Parasites. Parasites are those organisms which cannot live without the host, beta. Do you know that what is the causative agent of malaria? Plasmodium. Plasmodium is a parasite. It completes its life cycle in human and it completes its life cycle in mosquito. So it has two hosts. Okay. Some are the ectoparasites, some are the endoparasites, beta. Endoparasite live inside the body, ectoparasite lives on the body, outside the body, on the body surface. Okay, so parasite is getting the benefit, parasite is getting the food, parasite is getting, parasite is getting the food, parasite is getting the space to live into the host or upon the host. So these parasites can be ecto or endoparasites, beta. Ecto or endoparasites. So you understood predation and parasitism is same. But in predation, one will be called as predator and one will be called as prey. In parasitism, one will be called as parasite and one will be called as host. Beta, host is getting harm. Okay. Human when it is infected with the plasmodium, malaria, then in human only disease will be caused, na? So human is getting the harm. Alright, so host is getting the harm. When parasite comes into the host body, host will get the harm. Okay. Then coming to commensalism, beta, very interesting interaction. Commensalism, I will give you many examples of commensalism. Commensalism is an interaction in which one is getting benefit, one is getting the benefit and other is getting nothing, neutral. It is neutral, beta. Commensalism, one is getting the harm, other is neutral. One is getting the harm, other is neutral. I will be showing you many pictures and in those pictures you have to identify which interaction is this. Alright, so this will be a game now. This will be a game now. I will be showing you the picture and you have to identify which interaction is this. Okay, everyone? Okay, everyone? Are you ready? Are you all ready and excited? Have you all understood all these 6 interactions, beta? All these 6 interactions, what is the meaning of all these interactions? Have you all understood the meaning? Give a thumbs up. Have you all understood the meaning of all the interactions? Mutualism, competition, predation, parasitism, commensalism. Yes, everyone. Alright, let us start the game. Alright, everyone, please be ready. Let us start the game. Let us see who will give the correct answer first. Okay, let us see this. Okay, yes, everyone. Tell me, what are you seeing here? You are seeing cattle, cows. See, here I wanted to show you the cattle. I wanted to show you the cattle. Here, this is the cattle, beta. Cattle, na? Cattle. And you can also see this birds, beta, birds. Can you see these white birds, beta? White birds. What is this white bird? This is known as cattle egret. The name of this white bird is cattle egret, beta. So here we are showing the interaction between, this is the interaction between grazing cattle. This is the interaction between grazing cattle and cattle egret. And cattle egret. So, in the grazing cattle, see, grazing cattle is eating the grass. That is the predation. Between the grazing cattle and grass. What is this? Between grazing cattle and grass, this is predation, beta. Why? Why? Because grass is being eaten. So, grass is 0 here. Minus. Grass is minus and cattle is plus. Yes or no? Cattle is plus. Yes or no, everyone? Grass is being eaten, so minus, and cattle is plus. So, predation, you know, plus minus. Predation, you know, plus minus. Grazing cattle and cattle egret, beta. Grazing cattle and cattle egret. When cattle is grazing in the grass, na beta, some small insects are coming out. Insects. See, these insects are coming out. Insects are coming out. Small insects are coming out. When cattle is grazing, these are the small insects. Insects are coming out from the grass. And this cattle egret is eating the insects. So, grazing cattle here is not getting anything, but the cattle egret is getting the benefit. Yes or no? So, can we write grazing cattle is 0 here? Grazing cattle is 0 here and cattle egret is plus here. Whenever this type of interaction is known as commensalism. Commensalism. Yes or no, everyone? Tell me. If you have understood, please tell me. So, here I want to tell you grazing cattle and cattle egret. Cattle egret is getting the insects. Okay. They are not friends, beta. They are not friends. Okay. Cattle egret and grazing cattle are not friends. Okay. They are not talking about anything. So, simply whenever the cattle egret will see in the field grazing cattle, they will also come, because they know when cattle will be grazing in the field, insects will be coming out. And when insects will be coming out, we can eat those insects. Okay. So, their food is different. Cattle food is different. Cattle is not bothered. Okay. You can eat insects. I will eat grass. Okay. Here it is grazing cattle is plus, because here it is between grazing cattle and grass. But here grazing cattle is not getting any benefit of the cattle egret. Now it a bird. Bird is not giving any benefit or any harm to the cattle. What is the interaction between grazing cattle and cattle egret? Cattle egret is getting the benefit because of the cattle. Whereas grazing cattle is not getting anything because of the cattle egret. Simple words. Okay, everyone, let us go to the next question. Okay, tell me what is this and what is this? Can anybody tell what is this and what is this? Beta, this is crow. This is crow. This is cuckoo. Cuckoo. Or we call it koel also. Koel. Do you know? No, beta, no sparrow eagle. It is crow and cuckoo. Okay. So, have you heard that story? Have you heard that story that koel lays eggs in the nest of the crow? Have you heard that story? Yes, everyone. So, beta, this is parasitism. Parasitism. Okay. This is the host bird. Beta, this is the host bird. This is the host bird. And this is the parasite bird. This is the parasite bird. This is the parasite bird. This is koel. Koel does not know how to make a nest. Koel does not know how to make a nest. So, it will lay the eggs in the crow's nest. Okay. It will lay the eggs in the crow's nest. So, now there is similarity between the eggs of the crow and cuckoo. So, crow will not able to, not able to identify which are cuckoo's eggs and which are my eggs. So, the crow has to look after all the eggs. Okay. So this is known as beta brood parasitism. The name of this is brood parasitism. Brood parasitism. Here in this brood parasitism, what is happening? The cuckoo lays eggs. The cuckoo lays eggs in crow's nest. In crow's nest. Are you understanding, everyone? So this is an example of parasitism. Next figure. So you want to be understanding this. I know that. So let me tell you this. So this is goat, beta. Goat. And this is tortoise, beta. Tortoise. Okay. Tortoise. So there is a Galapagos Island, beta. Galapagos Island. On the Galapagos Island, na, only tortoise was living. So listen to my story. Only tortoise were there on the Galapagos Island. Okay. But then on the Galapagos Island, goats also came. Okay. Goats also came. So when goats came, na, then you know that goats have a very, fast grazing and browsing efficiency. They eat very fast. Goat eats very fast. Yes or no? Goat is eating very fast. It eats very fast. It eats very fast. And when goats came, na, they ate all the food. They were eating very fast. Tortoise was not getting the food. So that is why these tortoise, they have got extinct. This is competition, beta, competition. So on Galapagos Island, tortoise, the name of this tortoise is beta Abingdon tortoise. The name of the tortoise is Abingdon tortoise. Abingdon tortoise. Abingdon tortoise. Abingdon tortoise got extinct due to competition with goats, beta, with goats. All right. So this is competition is happening. Competition is happening, beta. Competition. Due to competition, the tortoise became extinct. This is competition. Bakri in Hindi. Yes, I know. Okay, everyone, coming to the next story. Yes, very nice picture. Let me tell you, these are the tentacles of sea anemone, beta. What are these? These are tentacles. Tentacles. These are the tentacles. Do you know tentacles? Have you studied that? Sea anemone. Yes, sea anemone. In which phylum you have studied sea anemone? I know you have studied animal kingdom. Tell me, sea anemone. Sea anemone. Yes, in Hydra also tentacles are there. So sea anemone also belongs to the same phylum. Coelenterata. Coelenterata. Yes, Coelenterata. Yes, and this fish is beta. This fish is clownfish. This is beta clownfish. Clownfish. So here these are the tentacles of sea anemone. Sea anemone. Tentacles of sea anemone. This is sea anemone, beta. These are the tentacles of sea anemone. And this is beta clownfish. This is what? This is clownfish. Clownfish. Here clownfish is getting protection. Clownfish is getting protection from the predators. And sea anemone is not getting anything. Sea anemone is zero here. So this interaction will be, which is this interaction? Commensalism. This interaction is beta commensalism. Commensalism. This interaction is commensalism. Plus and zero. Yes or no, everyone? Tentacles of sea anemone, because they are poisonous. Other predators will not come and they will not be able to eat the clownfish. Clownfish is getting protection in between the tentacles of the sea anemone. So sea anemone is not bothered. Okay, you can come. I don't have any problem. Sea anemone does not have any problem, but the tentacles of the but the clownfish is getting the protection. So you understood commensalism? Okay. What is this? What is this? Can anybody tell? What is this? Yes, it is veil. Yes, everyone, it is veil, beta. It is veil. And do you know that these small, these organisms are there, beta. What are these organisms? These are some kind of, arthropods. They are some other animals, beta. All these are known as, beta, barnacles. Barnacles. So these barnacles are growing on the veil body. Barnacles are growing on the veil body. Wherever veil is going, barnacles are also going. So barnacles are getting the benefit here. But again, veil is not getting anything. Veil is not getting anything from the barnacles. Not the harm, not the benefit. Not the harm, not the benefit. So again, what is this? What is this? This is also commensalism. This is also commensalism. This is also commensalism. Plus and 0. Commensalism. Next. Yes. You must have heard of mycorrhiza. You must have heard of mycorrhiza. Here, mycorrhiza. Yes, everyone. Mycorrhiza, mutualism. In the mycorrhiza, you can see that this is mycorrhiza. It is lichen, is fungi and algae. Here it is root of higher plant. You can see this root. Yes or no? Root of higher plant. Root of higher plant. And what is this? Fungi. Fungi. Root of higher plant and fungi, beta. Root of higher plant. Plant is doing photosynthesis, making food and giving food to the fungi. Because fungi is not able to make the food. Fungi is not able to do photosynthesis. Okay, but fungi is helping in increased nutrient efficiency. Fungi is helping in increased water efficiency, water absorption and utilization. So fungi is also getting the benefit. This is also getting the benefit and this is also getting the benefit. Both are getting the benefit. Mutualism. Both are getting the benefit. Mutualism, beta. Okay. Coming to the next. Yes. What is this? What is this? This is also mutualism. Here there is a orchid. This is an orchid, beta. And this orchid, one of its petal. This is one of its petal, beta. This is one of its petal. One of its petal. One of its petal. This petal. This petal resembles female bee. Female bee. So, the male bee will be attracted. The male bee will be attracted towards the female bee and male bee pseudocopulates. Pseudocopulates. Male bee thinks that it is a female bee. So male bee gets attracted towards the female bee. In that process, yes, it will do, yes, absolutely correct. Many of you have told absolutely correct. In this process, it will do pollination. It will do pollination. It will do pollination. And pollination is a kind of mutualism, but here the male bee is getting sexual deceit. Here the male bee is getting sexual deceit. So this is an example of beta sexual deceit. It is an example of sexual deceit. Do you know what is the meaning of deceit? Dhokha. Okay, sexual deceit, dhokha. So it is getting sexual deceit. Okay, and in this process pollination will be done. Pollination will be done. One more. This is also pollination. You know pollination. Pollination. You know pollination. Yes, the bee is there. You can see the bee. Bee is, when it is coming to take the nectar, it is dusted with pollen grains. The pollen grains will stick to its body, and now these pollen grains are there, and then it will go to the another flower and transfer these to the stigma. So transfer of pollen grains from anther to stigma. This is pollination. Cross pollination, self pollination. Pollination is an example of beta mutualism. Always remember, pollination is an example of mutualism. So I have told you, I have shown you so many examples of all these population interactions. One more is there, lichen. Have you heard of lichen, beta? Lichen is the association of fungi plus algae. Both are getting benefit. Both are getting benefit. It is also mutualism. It is also mutualism. So you can see on the tree trunk, this is a tree trunk, beta. On this tree trunk, you can see this white colored is lichen. This white colored thing is lichen. This is an association between fungi and algae. Algae can do photosynthesis. Algae can make the food. Fungi will get the food from the algae, and fungi will help in the absorption of nutrients. Okay, fungi will help in the absorption of nutrients. So this kind of relationship is mutualism. I hope between us also there is mutualism. Yes or no? You are getting benefit, I am also getting benefit. Okay. So yes everyone, that was the end of the session. So I have explained you all these 6 population interactions with the help of examples. So that is all for today's class. College will be starting in the 3rd week of May, I have told you. Okay. So I think this is my last class in the STAR program with you. Now from tomorrow onwards, another zoology teacher will take your classes. Okay. I hope you have enjoyed my classes. All right. So stay connected, stay tuned. Okay. And hit the like button, hit the like button, subscribe to the channel, and don't miss me. Okay. Don't miss me. Okay. See you soon. See you super soon in the campus. Bye bye. Take care. God bless you all. Bye, bacha. Bye. God bless you.